Okay, let's get started. So, hello everyone. I'm happy to welcome you to our monthly series of webinars with Stilla Technologies. My name is Justine Johnson. I'm a field application scientist with Stilla. Today, I have the pleasure of introducing our speaker, Beaven and Edelstein, sorry, every time. Beaven is a senior research associate with Kano Therapeutics and is a part of Kano's analytical development and synthetic biology team. He holds a bachelor's degree in human genetics from Trinity College, Dublin. Today, Beaven will talk to us about his work in precision QC in gene therapy. He's joined today by Christian Burns, who leads analytical development on the CMC team at Kano Therapeutics. He holds a master's degree from the University of Denver. He has honed his expertise in nucleic acid biochemistry, design, and manufacturing across the gene editing landscape at leading companies such as 4DMT, Mammoth Bio, and Chroma Medicine, leading various efforts from early drug discovery and design to process development and GMP manufacturing. At the conclusion of the talk, we'll open the floor for questions. You can submit your question throughout the presentation by clicking the Questions tab up on the right-hand side of your screen. It's marked with a question mark, and you can submit those questions throughout the presentation. So with that, please join me in welcoming Beaven.
All right. Hello, everyone. Welcome to my webinar. I, as Justine just said, am Beaven and Edelstein, and I'm going to be giving a talk kind of with us, Kano, and joining with Stilla to kind of show you how we use their instrument to do all the cool things that we do. So my talk, Precision QC in Gene Therapy, is going to go over monitoring DNA concentration and purity with digital PCR and the Stilla Nio instrument. So who am I and why should you trust anything that I'm saying? Well, I graduated in 2022 with my bachelor's in human genetics from Trinity College Dublin in Ireland, as Justine just said, and I joined Kano later that same year. And actually I'm coming up on 3 years with the company now. And I joined when it was really small. There were just 3 of us. I was the first employee. It was just me and the 2 co-founders, Floris and John. And over the years, I've got to work in a bunch of different teams, moving back and forth. And now I get to be part of this amazing team of 11 people working in process development and, as was said, synthetic biology and assay development. So our company started out knowing that we needed to quantify DNA. And we started where I think a lot of people do, with qPCR. So I got to develop kind of our qPCR assays from scratch, from the ground up, over the course of 2 years. And we got all the way up to 3-plex, multiplexed qPCR for looking at a bunch of different projects and a bunch of different targets. And over time, I got to troubleshoot and work with that a lot and see all the benefits and kind of the highs and lows of qPCR. I got to deal with amplification plots, which if any of you are familiar with qPCR, this is a pretty terrible one that was so bad I didn't know it was even possible, but I got to troubleshoot projects like this. And finally, in the past year or so, got to move on to digital PCR, which honestly was the best move that we could have possibly done for quantifying our targets. So in that time, I got to work with a bunch of digital PCR systems. I've used the Thermo Absolute Q, the Biorad QX600, and now we have the Stilla Nio, and we get to use it every day for a whole host of projects across our entire company. But today I'm going to talk to you about how we use it in our process development. So first, I'm going to tell you a little bit about Kano and who we are and what we do, and then I'm going to tell you how we use Stilla and the Nio to do all the cool things that we do. So we're a gene therapy company by name, and gene therapies have a couple of components. First, you have the delivery vector, and these are like LNPs. People are working on exosomes, endosomes, virus-like particles, almost anything you could imagine to deliver cargo to cells. There's a second component. There's the editing enzymes. These are your CRISPRs, your CRISPR-associated enzymes like Cas transposons, PACE systems, RNA bridging enzymes, almost anything you can imagine to edit DNA. And people are working very hard on these 2 things and making great leaps, even though they're very difficult. But there's this third piece of the equation. There's the actual DNA repair template that you use to insert therapeutic genes, and that's where Kano comes in. We have a special type of DNA repair template, circular single-stranded DNA, or cssDNA that we call it, and we use, and this is kind of what our company is all about. So why do we make circular single-stranded DNA? Well, we make it because cssDNA is really good. But don't take my word for it. I'm going to try and prove it to you. So cssDNA, when we use it for gene therapies in vivo, causes a very negligible immune response, even at very high doses. You can see here on the left, when compared especially with double-stranded DNA, it has very low cytokine induction, and there's very low immune response, leading to low toxicity, even at high doses of over a mg per kg in mice. And on the right, you can see activation of the STING pathway in primary human T cells. So this is one of the primary DNA sensing pathways intracellularly. Double-stranded DNA causes high activation and a bunch of toxicity, but circular single-stranded DNA doesn't. So it doesn't cause an immune response. But what can you do with that? Well, it turns out that you can get incredibly high integration rates without causing cellular toxicity. So we were able to achieve 60% site-specific integration of a 7 kb construct in primary human T cells regularly and repeatably. And we can see with very low toxicity, almost negligible, even at the highest dose of cssDNA. But we did see incredible deficits when using double-stranded DNA. And this lets us drop doses way down for kind of precise integrations and low off targets, or it lets us shoot the doses really high to get these incredible integration rates. So single-stranded DNA is really good, and it's really good at gene therapy, and that's why we want to make it. But there's an asterisk here. cssDNA is really good, but it is hard to make well. It's difficult to make cost effectively, it's difficult to make purely, and it's difficult to make in a form that's actually usable to get these incredible results. And this is what we do. This is Kano's pitch, and this is what Stilla lets us do. So I'm going to walk you through our production process and I'm going to give you 3 little vignettes where we use Stilla at each point, and we're going to show you data all generated by Stilla for each of these potential use cases throughout our entire process. So first off, we can use it in design and engineering. We have a polycistronic pathway for cssDNA production in bacteria that we can engineer using ddPCR. We then can take that and move it into production and fermentation. And then we can look at fermentation trends over time using the Stilla. And not even only that, but we can look at it downstream as well and see how our process can be improved and optimized. And then finally, we can use it for final formulation and release of our final product, including COA, QC, and anything else that you want. So first, let's talk about design and engineering. So this, I think, is where we get to use kind of the fullest extent of the Stilla's kind of powers. So we use 5-plex, one-step RT-CD-PCR to look at our engineered pathways. So that's a lot. That's a lot of words, but essentially we're just looking at the levels of 5 different RNA transcripts as we're engineering a system. And this lets us get data that you would typically need RNA-seq for, that would be incredibly expensive and take a long time to get, and you can get it really quickly and cleanly and do it kind of all in one, which has been really powerful for us. And it spits out data that kind of looks like this. You can see all of our positive and negative droplets across several different colors that we're monitoring all in a single reaction. And this lets us look at coincidence of different droplets if that's useful for us, and get all these different ratios for all these different RNAs that let us really engineer a system. So I'm going to give you a little example of what we did. We took this one system, system 1, and then we added an additional gene to this polycistronic pathway that was engineered and we believed would increase our yields of circular single-stranded DNA in productions. So you can see here on the left the yields and each different system that we used. And so each of these systems, 2 through 4, have this additional gene, and we engineered it with a small library of promoters to express at different levels what we believed would be at different levels. And we saw that several of the systems decreased yield, but system 4 actually increased it. Then we took our RT-CD-PCR and looked at what was actually happening under the hood. So on the right, you can see 2 components of the pathway, gene A and gene B, and then finally gene X, the additional engineered one that we added. So across all of the systems, you can see relatively consistent expression, except for gene X. And we saw that using stronger and stronger promoters gave us more and more RNA transcripts as we expected. And as the expression of gene X increased, we got a corresponding increase in the yield of our single-stranded DNA. But the really cool thing that we were able to see with this, which is always something you hear about, but you don't get to see very often, is that when we produced a lot of this gene X, so it's a stressful gene produced at a high level relative to a bunch of other RNAs, we actually saw global transcriptional repression of all of the other genes in the system. And we believe and saw kind of throughout the bacteria as a whole, which you hear about, but is really cool to be actually able to see. And in fact, we saw that if we increased the expression levels of gene X any higher, it actually caused a toxic effect on the cells and inhibited growth. So we're able to see kind of exactly where the limits are of our engineered system as we're engineering it kind of quickly and with very rapid feedback. So now we've engineered the system. We've engineered a production system for making our DNA. Now we have to actually make it. And what we can do is optimize our fermentation using digital PCR. We can take samples from every point along fermentation. So here's an entire fermentation process from left to right across about 2 days. And we can see the different yields of the different target DNAs that we're looking for on the right. So on the top, we have our blue triangles. This is our product, our circular single-stranded DNA. And then here's our contaminant, our host cell DNA. So these would be host bacterial cells that are lysing and releasing their DNA out into the supernatant where it can be measured. And then we also tracked the culture density, the OD600 over time, just to make sure that it was growing well. And so what this allowed us to do is that we can check through every step of fermentation when we're getting production, where we're getting production, and when it starts to plateau, because it will at some point. And so we can cut off our production exactly when we've kind of hit that balance point, and we're not just needlessly extending our fermentation beyond when it needs to go. And additionally, we can monitor cell health using this host cell DNA measurement. We can see when there are spikes in kind of host cell DNA that's been released into the media. And this lets us also time the fermentation so that we stop right before they overgrow and make sure that there's no unnecessary lysis. And you can keep the fermentation media as clean and pure as possible, even directly out of the fermentation. So making sure you're not putting any unnecessary contaminants in. And the cool thing is we can mark these and relate them back to other measures, which are even easier to get, like OD and glucose measurements, and we can kind of understand what's going on inside our process, not exactly in real time, but directly after the fact, so we can optimize it. So the next thing, it's really cool that you can do this in fermentation, but you can actually take this through an entire process. Because digital PCR is so robust to all kind of concentrations and levels of contaminants and inhibitors, it doesn't matter kind of what chemicals you're using, what buffers, what concentrations. You can monitor DNA throughout an entire production and purification process, no matter where it is. So here on the left is that same fermentation graph that I just showed you, but now it's compressed to show the rest of the processing all the way to the final formulation. And this is the first example and pass that we did almost a year ago now at this point, and we optimized from here. So you can see each unit operation and steps within those unit operations when you're purifying and seeing step recoveries at every single point. And you can also track contaminants such as host cell DNA and see where they're cleared, where they're not cleared, and what your final kind of concentrations end up being. And so you can substitute in and out parts of your process and just check immediately, like the right step recoveries that you're getting, what's going on inside the system, and if you're clearing contaminants, but maybe also clearing your product at the same time. And this has been incredibly useful for us to like rapidly work on our process. All right. And then finally, the final formulation. So I think actually this is the most interesting and unexpected result that the Stilla was able to show us. We had started to see lot-to-lot variability between our samples, and we were using kind of for our final DNA spectrophotometry, specifically the nanodrop, but we'd also used the solo VPE, which is specifically used in GMP processes, but agreed a lot with the nanodrop at times. So we'd use these spectrophotometry measures to look at our final formulation and what that concentration was. So usually you have only one way to look at what you're looking at. For example, in AAV production, you almost always use digital PCR to see what your titers are, to see how much you get out, because there aren't really any orthogonal ways of looking at your yields. But DNA, specifically purified DNA, there are a lot of ways to look at it. And the most common is probably spectrophotometry. And so we were using that. But we also compared it with the results that we were getting from the fermentation trends at the final formulation of our DNA with Stilla. And we were seeing some interesting results. So I'm going to walk you through this. There's going to be a lot here. But we took our circular single-stranded DNA and measured it via spectrophotometry and aliquoted all the exact same volume of this cssDNA. And we measured it, lyophilized it, and then we resuspended it to what we believed was 2 mg per mil. And indeed, you can see here, when we measure again by spectrophotometry and resuspend in water, we get this 2 mg per mil concentration value. However, what we saw was, depending on the formulation that we resuspended it in, so this is the same volume of liquid, and we used the right buffers that we were using to resuspend the DNA to blank our spectrophotometry measurements, we saw incredibly different results for how much DNA was in these vials when they were resuspended to the same amount. We saw in some of the most extreme cases a 30% difference in the expected yield and the actual yield that spectrophotometry was showing us. The amount of concentration that spectrophotometry read was almost as influenced by whatever liquid we resuspended our DNA in as by the actual DNA that was in the liquid that it was in, which was pretty crazy and pretty wild to us. And so we started to normalize by digital PCR, and we saw all kinds of other things that didn't make sense suddenly come together. In our electrophoretic assays, so in our agarose gels, we saw bands suddenly be the same intensity for our DNA. In cell assays, we saw integration rates suddenly make sense and match each other across different lots. And even visually, especially in some of these extreme cases where the concentration that Stilla reported was almost half of what the spectrophotometry said, we actually saw that the size of the pellets when you're lyophilizing lots of DNA was different. And the moment we normalized by digital PCR, they all became flat and normal. So essentially, we saw that spectrophotometry is almost a random number generator. If you care about how much DNA is in a sample, you really want to use digital PCR to measure that. All right, and that brings me to the end of the presentation. I want to leave a little shameless plug here, and I wanted to thank all of the Kano team, and especially Christian Burns, and also the Stilla team for making this presentation possible. And I also want to say that we here at Kano are open to collaboration. So if you are interested in circular single-stranded DNA, in checking it out and just talking with us, if you're interested in gene therapy, in ag bio, in DNA origami to make little dolphins that can move, there's some amazing papers you should read. You should really check them out. Or even just in trying out cssDNA and seeing what's available, please reach out to the email below. And I wanted to thank everyone for joining.
Fantastic. Thank you so much to Bevan for that great discussion about how you've been able to monitor DNA concentration and purity with digital PCR. So I'll throw out a couple questions. Please continue to input your questions into the questions tab. So first, you measure both very high and very low concentration samples of DNA in your final formulations. Have you had any difficulty with that dynamic range of measurement?
So no, actually, that was one of the really good benefits of digital PCR that we were able to find. It has an incredibly low threshold for what it can measure, even like single copies in, say, a 5 microliter reaction of DNA. So what you can actually do is, if you dilute down, you can take almost any concentration. And in fact, for a lot of our kind of final processing samples, we even use robots and other things to do serial dilutions, because you can take from like these were incredibly high, like 10 to the 17 concentrations, and bring them within range of the instrument. So because it's so sensitive, you can use kind of the entire range.
Fantastic. And in your experience, what is the benefit of using digital PCR over qPCR to observe fermentation trends?
Oh my gosh. So digital PCR is it's not nearly as well. One, it's absolute, so you get an absolute quantitation. You don't have to have standard curves, which are kind of have to be in a certain matrix. So if you have a standardized DNA that you're comparing it against to get your kind of quantitation, it has to be in a certain kind of buffer that will probably have an effect on kind of the result of your qPCR. And we saw this over time, really trying to optimize our qPCR. But digital PCR, you just get this one number out because the droplets are either positive or negative. But qPCR, you can't rely on your standard curve unless you've done a standard curve with the fermentation matrix, so with all of the acetate that's been thrown off and all of the cell proteins that are in there. And so it's just, there's a whole lot of junk in fermentation, and digital PCR, just you can dilute that away, but also just get your like yes or no whether it's worked and not have to worry about these fermentation, these like amplification curves, and just kind of get an actual result out without having to worry about it. And that's kind of the real benefit that we've seen, is you can put really dirty matrices into digital PCR.
Fantastic. And did you have to do anything special to optimize your assays to work with circular single-stranded DNA versus normal double-stranded DNA?
So actually, no, not really. So in PCR, all the strands separate and become single-stranded anyway when you're melting them. And we found with digital PCR that just will kind of offset you by a cycle, but it doesn't matter because you're running so many cycles. It still will measure if there's a copy or not of your target DNA in a droplet. This was, in qPCR, the bane of my existence, because in theory, it should shift everything by one cycle. You should melt your DNA, and you have, normally, when you have double-stranded DNA, you'll melt it, and you'll have your 2 strands, and then those will become 4. But with single-stranded DNA, your one copy would always be 1, and then the second cycle would be 2 instead of 4. So in theory, you should shift everything by a cycle to make everything match up. But in practice, that's anywhere between no cycles and like 3 cycles that it should be shifted, and it just made everything incredibly noisy.
And I think this next question kind of builds on that. How do you make sure that single-stranded DNA is not multimerized?
Not multimerized. So that is, so let's see if I understand the question. It's not like been concatenated into several pieces, like within a single strand, or that there are multiple copies of the DNA within a single one. I guess I'll answer the first one, and I can
Yes,
answer
single strand is the clarification.
So the single strand. So I think if you get, so you would only get if you have multiple copies of the same thing that have kind of been put one after the other. Digital PCR would always measure those, but as one piece of DNA, it would always measure those as one piece. So digital PCR is not the way that we have a bunch of other methods that we use. We have run a capillary electrophoresis and a bunch of other methods, HPLC and a bunch of other QC methods to look at kind of that and see the sizing. But digital PCR would just see that as you have one copy of your cssDNA. But we have other methods to measure if you get multimers.
And a follow-up question to the dPCR versus qPCR question. What is the sample that you add to digital PCR then? I assume it's cDNA or isolated DNA.
So we do isolate DNA from the fermentation media. For example, the like host cell DNA contaminant that I was showing, you just take the supernatant and spin out all of the cells, and any free-floating DNA which is dissolved in the supernatant, you can just take that and run that directly on digital PCR. You don't need to purify that any further. You can just dilute that down and run that. We found on digital PCR, and the matrix doesn't mess with it too much.
Yeah, and the same is true for the capsid encapsulated cssDNA. You don't need to do any downstream purification. The initial denaturation inside of the CDPCR takes care of all of that for us.
And you mentioned how digital PCR can be used instead of sequencing. Could you highlight some of these advantages?
Yes. So we've used it specifically We've used the RT-CD-PCR, the one-step, so reverse transcriptase CD-PCR. It works very similarly to RT-qPCR. You essentially just generate cDNA from your RNA transcripts, and then you quantify that. So if you have specific targets that you're looking for, the Stilla can do up to 7 colors, so you could do up to 7 specific targets. Or if you work with Stilla, you can multiplex them together to get up to 21 colors. You can look for all of these different targets, so up to 7 or 21 if you work with Stilla, and you can look for the amounts of these RNAs in solution. And it's both it has a wider range than sequencing and is more sensitive. So you might easily miss a read in sequencing, but you can find a single droplet in 20,000 that has your RNA transcript of interest. And it's also much faster. So instead of prepping the RNA specifically and sending it out and getting ready for sequencing or doing it yourself, you can just purify your RNA, load it directly into the Stilla, and 2.5 hours later, you have all your values and all your relative values, which I think is really powerful. And if you know what you're looking for, that can get you results really, really quickly and tell you kind of what you're looking for. That's how we use it instead of RNA-seq to kind of look at different RNA levels.
Great. Thank you for the very interesting and exciting talk. Do you integrate promoters with your circular single-strand DNA to alter the expression?
So the engineered pathway that I talked about earlier is separate from our cssDNA. So we use this polycistronic pathway to produce our circular single-stranded DNA, but it is not part of the DNA.
But for gene editing applications, there is a lot of times promoters integrated into there that when integrated into the genome act as the promoter for the gene that's being integrated.
Is a proteinase K or restriction digest step needed for digital PCR quantification?
No, we've found it's almost essentially you just kind of take your sample and dilute it to the range for the instrument and just run it. And we've had very few instances where that hasn't worked. Even with genomic DNA, which we've tested a bunch, especially with like measuring integration rates and other things for our gene therapies, we found you don't even have to cut up the genomic DNA, the human genomic DNA. You can just load that right in. It will be happily packaged inside the droplets, and you can just run that.
Perfect. And we can go into more detail on the next question as part of a follow-up, but how can you get 21 colors instead of 7? I did not follow.
Oh, yeah. So yes, you should all...
That's a question maybe I can take offline to discuss our universal reporters and crystal flux probes.
Yeah, you should totally check out the Stilla website. They have a little explainer there, but essentially you look for coincidence of separate colors, and you can look for coincidence of up to 3 separate colors, and that's how you get your 3 times 7, 21 colors. It's very powerful and very cool.
Great.
Yeah. Justine or one of the other Stilla folks would definitely be able to give you way more of a rundown of the nitty-gritty of how the crystal flex probes work than we would.
But the Neo has 7 colors base, so you can do just kind of directly 7 colors.
Okay, and with that, I would like to thank you both again for the wonderful presentation. Thank you to the audience for the wonderful and insightful questions. And with that, we will conclude the webinar.
All right. Thank you so much.
Great. Thank you, everyone.